Okay, the topic of this chat will be trying to compare something bizarre that I defined retrogiant to conventional retroviruses, so the way we conceive a retrovirus, because I believe that the retrogiant can challenge our current concept in retrovirology. In the beginning, I felt like Alice in the Wonderland. So we can't actually believe impossible things, but if we try with a little bit of exercise, we can believe as many as 6 impossible things just before breakfast. So before going through the retrogiant and something very bizarre, I hope you don't mind if I go and a quick look at retrovirus, so the way we conceive retrovirus. So we know from our medical school and over a century in the history of retrovirology that retroviruses are small, they have a small dimension, 100, 120, and they have a very simple backbone, GAG, POL, ENV, the reverse transcriptase, and 2 LTR. Sometimes retroviruses can be a bit more complex because we have a few accessory genes that complement that basic backbone. But despite being so basic, retroviruses are fascinating creatures, and we became interested in understanding their nature, especially for HIV epidemic. We understood the difference between a retrovirus and endogenous retrovirus, differentiate an endogenous retrovirus from a retrotransposon. We became even familiar with retroelement, like allele sequence in the human genome, and we became that clever to even cut and modify that basic backbone to produce a lentiviral vector to transduce cells in gene therapy. And we understood many things. We did many progress. But we failed at the sample in achieving an HIV vaccine because all the strategies and all the protocol were actually based on illogical assumption, and we failed. Anthony Fauci told us to stop, sit down, think, and go back to the bench. Now, some landmark events in the history of retrovirology. So we are talking and discussing about common retrovirus. So we all know Rous sarcoma virus in 1911, then cell transformation in vitro by Temin and the Italian Dulbecco. The concept and the discovery that Rous sarcoma virus had an RNA genome, and then the discovery in 1969 of retroviral oncogenes by Vogt, Martin, and Duisberg. Professor Duisberg was the heretic, is still the heretic professor who came to foolish interpretation in HIV epidemic, but is still the gold mine in retroviral oncogenesis, and he made a major contribution in the discovery of retroviral oncogenes. And then the fundamental discovery by David Baltimore and Temin, 1970, of reverse transcriptase, and this is something, a very landmark event because we could reverse the flow of genetic information. And then Bishop. So Bishop told us that for every gene you have in a virus, you have a mirror in the host. So host origin of oncogenes. And then in 1980, the discovery of the first human retrovirus, that is the human T lymphotropic virus associated to leukemia. And then the discovery a few years later of HIV by the French, it made, in part was made possible because of a contribution of Gallo interleukin 2 and the way how we cultivate cells. And 2003, the use of retroviral vectors in gene therapy. Few names, okay, few landmarks, names associated to retrovirus. And we could assist in a few years ago an interesting case of a genome invasion of a koala retrovirus in the germline, and this was just happening in real time. And we do have retrovirus, exogenous retrovirus. We have retrovirus as part of our genetic inheritance. Okay, a cat has retrovirus, feline leukemia virus. FIV, it is a lentivirus similar to HIV. But I was just thinking that dog, they don't have a retrovirus. Never, we could get the chance to isolate retrovirus in dog unless, some footprints in the human genome, in the dog genome, and might be humans be like dog. I don't think so, because it's too late. So at the end, okay, we came to this classification. So we did study Platon in the high school, and when we think of a man, we are all different, we are distinct. But when we think of a concept of a man, is one. So of course, when you discuss a retrovirus, not every retrovirus is like another one. So a feline leukemia virus is not like a human T lymphotropic virus. They have a different attitude, a different preference, different accessory genes. Okay? So we could group, and GAG is a group specific. So we could group retroviruses and classify retroviruses among families. But when you think of a retrovirus, what do you have in mind? You have a small dimension, GAG, POL, ENV, and 2LTR. This is the concept of retrovirus. Small dimension and a basic backbone. So my question was, are we missing something from that classification? Could we miss this one? Well, so the small one particle is at least 3 times larger than a retrovirus, and the large one is very massive. So you may be thinking, well, you must be joking. Why do you think that this is something giant, that giant, has something to do with a retrovirus? I think, it is actually quite impossible. And why we did not see over a history, centuries of retrovirology, at least 1 century, why we didn't see a giant, okay, in our retroviral people. How could this happen? Well, I think that it's very simple. It's really simple. I didn't do anything that difficult. I only tried a different fishing technique and a different bite. So when you think of retrovirus, you think small. And where do you go fishing? Deep. Deep where? Into the sucrose gradient. But if you suspect something different, you don't stay deep, you go up. Okay? That's very easy. And I'm sorry, in the era of sophisticated technique and software complex model, I think we should possibly go back and think, that being simple not always means being, not good enough. So what it is simple in science is beautiful. How did I reach that big particle and the small one? Okay, one day I decided that I could use an anti-feline leukemia virus, that is a conventional retrovirus causing leukemia in cell, to stain some human tissues. Cat tissues? No, human tissues. So I used an anti-FeLV to stain human tissues. So I took a little bit of brain, a little bit of liver, kidney, lymph node, and I start, my painting with my monoclonal antibody. Then I looked at the microscope, and I saw that my antibody, my monoclonal antibody, anti-feline GAG, was actually labeling some structures, unusual. in tissues. So the first thing that a nasty person will tell you is artifact. That's the first thing, because they have a possibly pessimistic attitude in science. I didn't bother that much. I had some knowledge, vague knowledge on retroviruses, and I liked these spores flowering in the wind, so I sent everything to mass spectrometry. So mass spectrometry is still in its infancy. It's a bit delusional, but can guide you. So I was working on human tissues, whole human tissues. I had a list of human peptide. That's easy. But in this pool, I had retroviral peptide, feline related, and I was using an anti-fel. And I had an histone 4 that had the same footprint of a giant virus. And this was for me a big surprise, because I didn't know that the virus could be giant. And I knew that while we do have a variance for the histone families, we do not have variant on human histone 4. So we only have one histone 4. It is very conserved. You can see here the histone variants for the other histone family, but not a variant for histone 4 except post-translational modification. So I got a little bit suspicious. And then this guy from Canada, he came back to me and he told, listen, I can see that this footprint is unique for a virus, because I could not find this sequence in any human database. And look, I was working on human tissues, and I had my human histone was there. But along with my conventional histone 4, I had, this unusual histone 4 with a viral footprint of a giant virus. And then something clicked in my mind. I looked at the giant. I didn't know they were giant. They were giant virus. I was missing this part because I come from a retrovirology background. And I looked at my spores flowering in the wind were very actually kind of suggestive of a giant mimivirus in the amoeba. I did try a gram staining on, the human tissue to just see that I could get the same granules of a giant virus in the amoeba. So I was running after a classic retrovirus with my anti-feline leukemia virus, but something was pointing to a different direction. I was working on tissues, but I had really to solve a big problem. I had to isolate the virus, if really present. We didn't know. And verify if we had genetic material inside. In the era of metagenomics, I choose the simple, old sucrose gradient. But I picked from the shelf an old paper from PNAS by Professor Gallo, where he did describe the discovery of the first human retrovirus, the human T lymphotropic virus. But as I told you, this time I had a different strategies. All the school retrovirologists think of a small dimension, and I was suspecting something giant. That's pretty much what I have done. Not very, this work is really preliminary, but this is what I have done. Before I was working on human tissues. This time I took the human T-cell leukemia, because they are cancer cells, they are T cells, And I did remember at the time of HIV discovery was a big fight, because no, I gave you the interleukin-2, or you wouldn't be able to grow and isolate without my cell model. So we had already transformed cell line, and we could place in culture, harvest the virus, and do everything we could. So this time I did a sucrose gradient. I took the fraction, and sent to electron microscopy, a bit of gram staining, and reverse transcriptase activity. That was, clear in Gallo paper that they did the reverse transcriptase activity, okay? And to prove the concept, first a pan-retrovirus PCR, and then the whole genome sequence. In this picture, you can understand that my idea was is already clear, because I asked for a gram staining, and I asked for a reverse transcriptase activity. And I asked to amplify this fragment of the reverse transcriptase. It was clear enough to me, okay, that the giant could be something to do with retrovirus. Old protocol. This is, kind of old technique, where if you use a semi-nested approach, you will be able to amplify a segment in the reverse transcriptase enzyme. And I did find the band. It is 150, 180, okay? And you can amplify a very conserved segment of the reverse transcriptase over the retroviral species. And then I just compare the reverse transcriptase activity of my viral lysate to just a commercial kit. And this is the whole genome sequence, okay, the functional annotation. So what you see from this giant, this mega genome, mega base, is that you have, you go through the functional annotation. I'm not an expert in bioinformatics. I'm only an immunologist, and, but people working on understanding the functional annotation, so we did notice that this big giant had genes in common between archaea, between eukarya. There was a virus apparatus, viral proteins, but this is just nothing. This is, at the end will be more interesting. And of course, there was inside the reverse transcriptase. So I got a preliminary comment, but this man, he helped me in understanding the retroviral nature of my particle. So he is professor in Harvard, very expert in feline retrovirus, and he told me, no doubt in my mind, your findings will challenge stuff all over the place, and not just in the specialized retrovirus field. Many will simply say, this stuff is so gigantically big, so why haven't we all seen it before? That was my first reaction, and if you hadn't been using my monoclonal antibody to mark the inclusion, then I mostly likely wouldn't have given the rest of it a second thought. Sorry to be blunt, but incredulity will be most people's first reaction. Opposition from old school established retrovirologists will be a relatively trivial secondary consideration. I am both shocked and amazed by your findings, but I imagine we can all agree that no matter how expert we are as immunologists and virologists, endogenous viruses remain a black box to us all. But please, to satisfy my curiosity, why did you use this monoclonal antibody? This gentleman, he came across the paper, the one I tossed in the platform, And he was very surprised because the antibody I did use and purchase from Santa Cruz, he made. And he is the one who made the hybridoma back to the 80s. And he told me something about, how to further expand the thinking of this unique retroviral antigenicity. So I have isolated, before we were on human tissues, this time we were on cell, and I isolated the retrogiant from acute T lymphoblastic leukemia established from a PBMCs of a 14-year-old Japanese boy with an acute lymphoblastic leukemia and thymoma, a diagnosis in 1973. It should be noted that usually these cells are screened by PCR, and they are HBV negative, hepatitis B negative, C negative, HIV-1 negative, and very surprisingly, human T lymphotropic virus negative. And Japan is the crib of the human T lymphotropic virus. Yet, I could not find by PCR the human T lymphotropic virus as in this cell. So I was pretty much confident that I didn't have confounding factors of archetypal retrovirus in my sample. And the retrogiant had a unique and distinct retroviral antigenicity, FEL-related. One hybridoma, one monoclonal antibody, was made from an hybridoma against FEL-GAG. It is the core protein, and it is GAG specific for feline leukemia retrovirus. Not every retrovirus is like another one. This is very important in our understanding. Somebody told me, you really need to observe more. Because yes, you have the genome, but you have to put together that genome to your particle. And we didn't have any problem actually to do additional sucrose gradient. The cells were there. And we observed more. And now I'm sorry, I will show you something bit. So what I did disclose in the platform was only, a small particle, 400, and there is also this big one. Not an archaea. Polyploid. The same scenario, the small one and the big one. Both giant. Both labeled, the big one and the small one. Now, this was on the sucrose gradient that we did from the cells. Nothing else was there. The more you look, the more trouble you find. So not only the predominant form of a particle 400, but something that could be 800. Now you may be thinking, you maybe only discovered an archaea then. So archaea, they are big. These particles have the strong archaeal features. And we know now with all this bioinformatic test, the archaea, they can have both RNA and DNA. And in my functional annotation, I did see many enzymes of the archaea. And maybe I only found possibly an archaea or something similar in human T-cell leukemia. This is possible. So, in archaea, they do have reverse transcriptase activity. Even bacteria, they do. But, yes, they do. You know, bacteria, they have the reverse transcriptase. But do they have an envelope? I don't know. I'm not an expert in archaea. But do they have, viral oncogenes? And where is the 16S ribosomal RNA? I couldn't find. And do they have a retrovirus inside? And you may be thinking, no. Why do you think it's a retrovirus inside your giant body? Gag and Paul is not enough. I don't think I will impress anyone telling I went with an anti-gag, the large particle as positive for gag, and you will tell, well, you don't prove anything. Could be, a retrotransposon, could be a reverse transcriptase, archaea. We are finding out that they have, the reverse transcriptase. Well, I don't know, not sure. But the problem is I did not only use an anti-gag. This was too simple. I think the most important antibody I did use in the context of my work is an antibody that we call C11D8. And this antibody, C11D8, is extremely known in the field of feline leukemia virus. This antibody only recognizes this epitope, MGPNL. And it is found only on the envelope of a FELV and nowhere else. It is a biological marker, and no one can really question the specificity of this antibody, because everything will fall apart. Never this specificity has been in question, and this antibody, anti-MGPNL, is the distinction, because this epitope only belongs to FELV envelope, including not only the exogenous FELV, the A1 particle, including endogenous FELV element, of endogenous retroviruses. So I could understand possibly, because of the presence of this epitope, and the literature, and all this, the history of this retrovirus, that we could, you see many paper, and this is the most important. So you go with an anti-gag, you won't prove anything. You will go with this antibody, you might be prove something else, So not a generic transposon, a gypsy, or anything, but kind of pointing to the endogenous nature inside this giant particle because of this epitope. This epitope is unique for FELV. So how did my interpretation? Of course, this is very preliminary. I worked in difficulties, so it was actually, very difficult to reach that point. But I have this giant, this unique retroviral antigenicity. I have, the sequence, the full sequence. Then, I decided that I could go back and study themin again and something. So if you see the functional annotation of the giant, you can see that there are genes in between the 3 domains of life and plus a retrovirus inside. So I thought that possibly, this is a unicellular organism because it is a cell. It is a cell independent from the host. Could be something maybe having a real phase to that speculation around Luca and around, the shift between the early RNA world to the modern DNA world. And themin was the one suggesting an ancestor for the reverse transcriptase. And I think, that possibly if we read Temin carefully in his Nobel address, is actually telling something. So Temin is much more complex than David Baltimore, because Temin did not only discover and purify the reverse transcriptase, but Temin came with the provirus hypothesis. And Temin, he was the one suggesting an ancestor for reverse transcriptase. But it's actually Temin, before getting through this discovery, he was also very busy in cell transformation in vitro. He knew all the process leading to cancer formation. And he said, when I speculate on the origin of Rous sarcoma virus, I can see that Rous sarcoma, so the cancer, appear before Rous sarcoma virus. More specifically, other events, not including a virus, led to the formation of a gene for cancer and chicken sarcoma. It's telling something in between these lines. But I am not really interested in finding an age for this unicellular thing that I found in human T-cell leukemia. Might be possibly fit this paradigm, but phylogeny, I assume, they can explain many things. How we can explain this with a tree? So Temin again, you have an avian leukosis virus for a random recombination in the cells. Just random gives origin to Rous sarcoma virus. This is how Rous sarcoma virus is originated, by a random recombination process between a cancer cells and avian leukosis virus. We can't explain this with a tree. So it is actually, okay, so my point is understanding the nature, the retroviral nature of this giant, because they are so evident, they are there. The meaning of this retroviral nature, if really there is a retrovirus inside my giant, and understanding if this something I called retrogiant, because in my opinion has something to do with retrovirus, are dangerous but enough, smart enough not to release danger signal. So I think we can think of Polly Matzinger. She's one of the most brilliant immunologists. She started being a Playboy bunny, and then she became professor. But she is one of the immunologists that can actually tell you why you don't reject cancer. Not because of self and non-self. So we are taught in our medical school, you reject what it is non-self. So, but we don't reject cancer, and cancer is non-self. And we do not reject ourselves during puberty. And we do not reject, we can't do a vaccine if you just inject a peptide. So you must release danger signal. Otherwise, you won't prime an antigen-presenting cells, and you won't prime an immune responses. So I think that giant, the big one and the small one, if you go through the list of prediction, everything is prediction, but everything is possibly pointing to something. I did use an anti-FeLV. The anti-FeLV marked the inclusion. And look at the list of predicted oncogenes that we have inside this mega virus, mega thing. Okay? So, again, the fusion protein, FeLV related, and look, the sarcoma, feline sarcoma virus, that is a defective retrovirus that needs the envelope, feline envelope, to go together and replicate, and many GAG fusion proteins. and many transforming, and tyrosine kinases, and the viral notch. This is a bit complex here. Now, this is PICS. The top hit peptide is a tyrosine kinases of sarcoma, feline sarcoma. So we have oncogenes inside. I speculate we have oncogenes and many, not only one. So if you go back to a retrovirus, which one will give you cancer here? Which is the gene? No one. They're all genes for replication. How they can become dangerous? Because they can possibly integrate. And they can activate a proto-oncogene. Okay? But there is no danger in that simple backbone. You see, an avian leukosis virus is not dangerous. It's only slow transforming because it doesn't carry oncogene per se. But a Rouse's sarcoma virus is very transforming because it has an oncogene. So if we think then, we have transducing oncogenic retrovirus, highly oncogenic, and then even the human T lymphotropic virus that so far is the only retrovirus associated to leukemia is actually very mild. Because, yes, TAX is not an oncogene. TAX can bind the NF-κB transcription factors and transactivate something, but it is not, doesn't give cancer. And the retrogiant could be this retrogiant harboring many things. Proteins, they harbor proteins. They harbor oncogenic proteins. And all of these are oncogenes. And so, at the end of this little chat, we add. So, we have, on one hand, retrovirus, small virus, Gage-Polanenv. Of course, the provirus inside the cell, that's obvious. And then, sometimes they can be dangerous, sometimes they are not. They're even useful. Retrogiant, on my hand, are big entities having a synthesis of archaea, bacteria, and eukarya, a large genome, a retrovirus inside related to the feline lineage, and many transforming oncogenes. So, the synthesis is this, the way I see. That's something bizarre I called retrogiant. I understand this work is very preliminary, but if you see at the big particle and the small one, and the functional annotation from that particle, you understand the timing is here. You understand concept of a cancer and epigenetics through the histone, not only JAK and STAT and beautiful signaling pathways. And you understand the archaea feature, giant virus and eukarya is there. And this, I think, concept wouldn't be possible without the discovery of the giant virus. And this is possibly digging into the cross-species transition of retroviruses. And look, retroviral oncogenes, and we have many inside the giant. And Bishop with the concept that you have a retrovirus and you have the provirus. So host origin of oncogene, but what is the shuttling system? What is the shuttling system? So I think, in my opinion, that this unicellular organism, okay, has something to do with our epigenetics, is shaping our epigenetics, and I think will impact the IFLIC number. So I think we won't be able to reach the IFLIC number if we should actually find, and I think will be possible to shift to the IFLIC number if we have, if we can target this, some neutralizing epitopes present in that giant. So the works I think that needs to be done is, of course, you don't have to believe me because I'm not a prophet delivering a sermon, but it is actually, we have cells, so we can go back to the bench, we can isolate the giant, okay, and we can go for extraction and define better the genetic anatomy, and then see, okay, validate a diagnostic test. I think, the GP27, GP70 for the retrogiant works very well because this was the bite that kind of permitted the discovery, and then understanding concept of integration, then, see if you see that virus in the cell as well, okay? Understanding concept, how this giant can impact, so the, evolution, the pathogenesis, and what can be the role of the large particle in determining, the, disease. I think it's done.
I think, as I told you, I'm not sure that I'm convinced that this is a retro giant virus. But you may be right, from as much as I understand so far, in having a giant virus carrying, a sequence of a retrovirus that may transform the cells that is infected, rather than integrate it in cells. So,
yes.
Which,
yes.
Of course, is it's much less.
I think that's the point. Yes.
Yeah. So I think it's very possible that in giant virus in general, we see what you have. So they, of course, there is no tree anywhere. So there are mosaic of genes, and even if you're looking carefully, we just came out with a paper with Philippe. If you're looking carefully in the gene, usually even the gene are mosaics coming from different origins. So they pick up sequence.
Pickups, yeah.
And of course, if they are infecting a cancer cell, they may well be able to pick up genes from cancer cells.
Yes.
So it's not surprising. I mean, it's something that is not so hard to believe. And based on this, of course, if they transcribe these genes, this may well be infecting.
So it is, expression of the protein giving, the disease. Yes, I think so.
Yes, it may be a part of the sequence. The big virus may be a carrier of oncogenic sequence that may be integrated.
Yes.
In the genome of the host. This is possible. This is interesting. This is possible.
Yeah.
And will not surprise us because as far as we've been looking, all the viruses that we've been seeing, the giant virus, are all composed by genes of different sources, archaea, bacteria, eukaryotes, mostly unknown origin, and even the genes themselves are including part from, we get some sequence with viral and bacteria origin in the same gene. So it doesn't surprise me that it pick up sequence. It doesn't surprise that it gives sequence. So I think the really next step is that, and I think we'll be happy to discuss that with you, is to help you to do what we know to do on virus, not on retrovirus, but on virus. So really comparing that to any other, analyzing the genome with you very carefully to see what is on and what is in, and really doing tree, even if you don't like tree, with topoisomerase and DNA polymerase and the RNA polymerase and so on that we are very familiar with, and help you to fix that out in where is the skeleton of this thing. So, and of course, what's emerged now as a concept in giant virus is that there is a small skeleton, like the ribosome for the cell, and around this skeleton there is 100 of genes of different origins that traduce the past life and their adaptation to a specific medium or a specific cell. So all are different, even in the same species, but all get this common architecture on really transforming RNA and DNA and so on. So we can help you to do this if you're.
Thank you very much. Thanks.
A very simple question. Okay, the Paul, the RT, the Paul. Did you make the phylogeny with Paul? Because you show very nice phylogeny at the beginning.
With Paul? No, I didn't. No.
Because you will see if it's ancient, not ancient, and if you group with.
Yeah,
so only
that could be done.
This phylogeny first, and then the other.
Okay. Okay.
Question?
Okay.
No, it is interesting. The last thing is that because you find that by chance.
By chance, yeah.
It is interesting because you find that by chance. After that, as soon as you get a theory, it's become dangerous. But as long as you find things by chance, it's true. And when you start to make a theory, it's not true. It's so true. Do you understand what I'm saying?
Absolutely, yeah.
So you start to bias your analysis when you get an opinion. But when you just find this and say, oh my goodness, what
just
it is.
you don't have a prejudice, and that's fine.
Yeah.
This will help you in interpreting because you don't have that much.
Yeah, but definitely we're interested in trying to analyze.
Thank you very much. Yes.
And put that in the word of giant virus, no retrovirus, but giant virus. And you look after this if this thing can be transferred, the retrovirus part and the Oncogenes part. Could it be transferred to human cells? That's not a part.
Well, but that's will be an important, step. Yeah. Okay. Thank you.